Hello everyone, I am Anju Giri. I'm a postdoctoral researcher at Cornell University. Today I'm going to talk about getting haplotype expression around the gene and its utility in the improvement of genomic prediction of complex traits. We all know that gene expression can be as a result of mutation in the cis or trans factors. The cis effect in mutation could be due to the variation in the region right around the gene, whereas trans could be arising from anywhere in the genome. In this study, we get cis expression from the gene region to know how much of the expression are as a result of element right around the genes. And since these cis haplotype expression are less sensitive to environmental perturbation and genetic context of the individual, we hypothesize that the partition of expression from the gene region would be more transferable compared to the major expression values. So in order to test that, first we partition cis and trans expression using haplotype relationship metrics. The haplotypes were defined based on the polymorphism in the gene. For example, here the taxa2 and taxa carry same haplotype in gene X. And in this way, the haplotype relation matrix were created for each gene as a control for cis, and all those metrics were combined to get trans effect. And then we fit a mixed model for any gene for all the tissues expression data that was previously collected in the lab, which includes germinating root, germinating shoot, leaf-3 base, leaf-3 tip, mature leaf tissues, and kernel tissues. So once we have these haplotype expression values for each of those tissues and genes, we wanted to see how the correlation distribution looks like with the major expression values. This different panel here represent different tissues correlation distribution between major and haplotype imputed expression values. And as we can see in here, the haplotype expression are moderately correlated with average correlation ranging from 0.42 to 0.46 across these different tissues. We didn't see much variation in this distribution across these tissues. But we are more interested to see like how the correlation would looks like across these 7 diverse tissues, because we had hypothesized that the haplotype expression would be more transferable across the tissues compared to the major expression values. As we can see that the correlation across tissues for the haplotype imputed expression, which is this line in coral here, has higher correlation compared to the major expression values that this peak around 0. So partitioning out the trans component that includes more developmental and environmental noise in expression, we got higher transferability across the tissues for haplotype expression. So after getting this cis haplotype expression, we looked at the genomic prediction model to predict 26 complex traits across 2 diverse populations in Maize, which includes a nested association mapping panel with 5,000 individuals belonging to 25 families, and a Goodman Association panel, which is a diverse panel of 280 individuals. The model was trained and tested in either of these 2 populations. And before actually using the haplotype expression values, first we established the baseline for each trait using 100 randomly permuted expression values of haplotypes. Because the haplotype structure itself results in a moderate prediction accuracy, and we wanted to see like how much gain in accuracy we can have using the haplotype imputed expression values, which is this portion of distribution towards the right. When the model was trained in Goodman, which is a small subset of diverse population, and tested in NAM, we saw consistent increase in accuracy. However, the increase in accuracy was higher when the model was trained in NAM and tested in Goodman, which could be coming from the larger sample size in NAM, which includes 5,000 individuals, and less diverse haplotypes present in NAM. It has just 25 families in there. Overall, we saw 14% improvement over random expression values. However, this percentage improvement depends on the trend, on the trait and training population. So using this cis haplotype expression values, we are seeing gain in prediction accuracy over the random. And with this, I'd like to thank my lab members, my funding source, and happy to take any questions. Please feel free to reach out to me at this email. Thank you.